Carbohydrates. Today's topic is carbohydrates are widely distributed in plants in which they are formed from the carbon dioxide of the atmosphere and water by photosynthesis. Plants use carbohydrates as the precursors of the synthesis of protein, lipids, and other organic compounds. Animals obtain their carbohydrates from plants. Definition. Carbohydrates may be defined as a polyhydroxy aldehyde or ketones or compounds which produce them on hydrolysis. Carbohydrates are the polyhydroxyaldehyde or ketone which at least three carbon atoms and an active carbonyl group, which may either be aldehyde or ketone group. They also include those compounds which yield them on hydrolysis. Functions. Carbohydrates have a variety of functions in animals and human body. They supply energy for body functions and for doing work. They are a structural component of many organisms. They exert a sparing action on proteins. They provide the carbon skeleton for the synthesis of the some known essential amino acids and fats. Some carbohydrates are present on the tissue as tissue constituents. Starch forms a main source of carbohydrates in the diet. Classification of carbohydrates. Classification of carbohydrates में आता है monosaccharides, disaccharides, oligosaccharides and polysaccharides. This is a table flow sheet. You see this. So next, polarimeter is an instrument by which the specific rotations of the optical isomers are detected. Expression of the optical activity. Optical rotation to the left, such as lever rotation, is expressed with the signs of I negative. When rotation to the right, such as dextral rotation, is expressed as D positive. Recipe mixture. When equal amount of dextrorotatory and levorotatory isomers are present, the resulting mixture has no optical activity since activities of the isomers cancel each other. Such a mixture is said to be a racemic mixture. Resolution. The separation of optical active isomers from the racemic mixture is called resolution. Mutarotation. When the aldohexose is first dissolved in water and solution is kept in optical path and plain polarized light is passed, the initial optical rotation shown by the sugar gradually changes until a constant fixed rotation characteristics of the sugar is reached. The phenomenon of a change of rotation is called mutarotation. The mutarotation is due to the existence of two optical isomers of glucose, namely a D-glucose with a specific rotation positive 112.2 degree and B D-glucose with specific rotation plus 18.7 degree. Alpha and beta isomers are called anomers, and the carbon atom responsible for this is anomeric carbon atom. Anomers are isomers differing in configuration of a particular carbon atom alone. A freshly prepared aqueous solution of alpha D-glucose has a specific rotation of positive 112.2 degree. With this solution is allowed to stand, the rotation falls to 152.7 degree and remains constant at this value. This gradual change in specific rotation is called mutarotation. This is the structure of D-glucose and D-gluconic acid. Tolerance region लगेगा इसमें. Reaction with alanine, the aldehyde group of glucose condenses with amino group of alanine to form the Schiff's base. Fructose also gives Schiff's base with alanine. The browning reaction occurred during the baking of bread. Another mixture of carbohydrates and proteins is believed to be due to the formation of Schiff's base between the amino groups of protein and the aldehyde group of carbohydrate. Oxazone formation. An important reaction of the reducing sugar monosaccharide and disaccharide having potential aldehyde or ketone group is their action and the phenylhydrazine to form phenylhydrazones. Reaction with the phenylhydrazine involves only two carbon atoms, namely the carbonyl carbon atom and the adjacent one. The steps can involved in the phenylhydrazine reactions are one molecule of glucose condenses with a one molecule of phenylhydrazine to form the soluble glucose phenylhydrazone. In the presence of excess of phenylhydrazine, another molecule of phenylhydrazine enters the reaction. According to Haworth, glucose exists in the six-membered ring structure called the pyrenoform, and fructose in five-membered franoform, glucose or fructose. Disaccharides are the sugars containing two molecules of monosaccharides. Disaccharides are formed by the condensation of the two molecules of monosaccharide with the elimination of one molecule of water. In disaccharide, monosaccharides are linked by the glycosidic bonds. The properties of the disaccharide depend to great extent on the type of linkage. If the two potential aldehyde or ketone of both monosaccharides are involved in linkage, the sugar will not be exhibit reducing properties and will not be able to form osazones such as sucrose. But if one of them is bound in this way, it will permit reduction and osazone formation by the sugars such as lactose, maltose, which are also known as reducing disaccharides. देन फर्दर उसका माल्टोज और आगे से उसके लैक्टोज मेटाबॉलिज्म वगैरह उसमें बताई हुई हैं, वो ऐसा ऐसे डिटेल में पढ़ेंगे इसमें.